Moreover, it's a broad term used for those species which not only eat other species but 2. Introduction. Different species interact with each other to compete for food or space through predation. Predation is called as biophagy and defined as one species feeding on other living organism. Moreover, it's a broad term used for those species which not only eat other species but often involves in killing of other species. On Earth, as ecosystem, humans are the major predators. It is an association where mostly stronger attacks the weaker organism. However, considered more broadly, predation can be distinguished into 5 specific types, namely, herbivora, parasitoidism, carnivory, cannibalism and parasitism. Herbivora is a type of predation where all type of grazing animals prey on plants. This type of prey-predation association does not involve direct killing but plants, seeds or fruits eaten by animals causes damage to the green plants. Carnivory is a typical kind of predation where predator or stronger animal prey on other carnivores organisms or on herbivores. Another specific type of predation includes parasitoidism where an organism called parasitoids, generally these are insect parasitoids, predates on another insect's called host insect by laying eggs near or on the host insects. These eggs, hatched into larvae that feeds on tissues of host insect and subsequently killed and eaten by the insect parasitoid. Parasitism is a type of predation where parasites, plants and animals, are the organisms that do not kill and consume the prey or hosts but depends on them for nutrients. A special form of predation is called cannibalism in which predator and prey belongs to same type of. 3. Species. Certain groups of tribal people show cannibalism where on death of any family member they eat their body parts. The predator does not interact with prey only but with other predator species through competition. The interaction-mediated competition between predator species is indirect. When the prey of different or same species competes for food and space among themselves creates indirect competition between predators. Whereas when predator compete for same prey species i.e. in short supply it causes indirect competition between predators. Fig. Point 1. Predator 1 Predator 2 Down pointing arrow direct interaction, indirect interaction. Down pointing arrow Prey 1 Prey 2. Predator 1 Predator 2. Prey. Figure 1. Direct and indirect competition in a predator-prey system. Predation is considered as a useful interaction when animal predates on pest as prey but predation is considered as undesirable when prey is a valuable resource. Predation is an important interaction that reduces the abundance of prey and restricts its distribution. Thus, predation secondary to competition is the main type of interaction that influences the community shape, structure and organization. 3. Models of predation. Organisms use 2 types of system called discrete generation and continuous generation systems by following 2 simple types of prey-predator models. 4. Model for organisms with discrete generation system. Discrete generation system is used to explore prey-predator interaction models. Several insect hosts act as prey and insect parasitoids acting as predator under seasonal environmental conditions have 1 generation per year and are described by following type of models. Logistic equation describes the increase in the prey population in the absence of predators and given as NT plus 1 equals 1b NT NEQ NT. Where NT equals population size, T equals generation time, B equals slope of reproductive curve, and NT NEQ equals difference in the population size from equilibrium size of population in absence of predator. We must subtract a term related to the number of preys eaten by predators from the logistic equation of prey under the absence of predator. The logistic equation of prey in the presence of predator is represented as NT plus 1 equals 1b NT NEQ NTC NTPT. Where PT equals in generation time T, the size of predator population, C equals constant determining the predator efficiency. Assuming that the predator's reproduction rate depends on the population size of prey, the equation for predator population is given as PT plus 1 equals QNTPT. PT equals predation population size, N equals prey population size, T equals generation time, and Q equals constant determines the prey utilization efficiency of predation for its reproduction. When these equations are put together and interpreted after calculating them over many generations resulted into a cycle of prey and predator numbers. Fig. Point 2. There are 4 possible outcomes of these calculated equations. A. A stable equilibrium without oscillation. 5. B. Divergent oscillations, causing extinction of either prey or predator circa. Convergent oscillations, causing extinction of either prey or predator. D. Stable oscillation. Figure 2. A hypothetical classical oscillations in prey predation system with discrete generation depicting population changes. X-axis depicts the population size, Y-axis depicts the time period. Continuous generation system. Organisms with continuously occurring births and deaths have overlapping generations such as several prey and vertebrate predators provide good examples. The continuous generation system used by the predator and prey interactions is described in an independently derived set of mathematical equations given by Lotka-Volterra, 1926. Lotka-Volterra predation equations. In 1925, a physical scientist and mathematician, A.J. Lotka in his work, Elements of Physical Biology, suggested the early and very first model of predator-prey interaction system. Independently, Volterra developed a similar model of predator-prey interaction in 1926. The model assumed that the responses of predator population and prey population are proportional to the product of predator and prey population size. The predator-prey equations given by Lotka-Volterra describe the interaction between prey and predation as a dynamic. 6. Biological system. The predator and prey populations follow a nonlinear, first-order differential equation and represented in pair of equation as dx dt equals x beta xy di dt equals delta xy gamma y. Where x and y is the number of prey and predator population and dx dt, or di dt, are the growth rates of prey and predator population in time, t. The symbols alpha, beta, gamma and delta denotes the real and positive parameters related to the interaction between prey and predator species. Lockable-Terra predation equation is based on the assumptions. 1. Predator can eat limitless. 2. Supply of food resource i.e. prey depends on the prey population size. 3. The rate of change of population directly depends on its size. 4. Environment is constant, inconsequential genetic adaptations for both species. 5. All-time unlimited food supply. Lock the predator and prey. The symbols alpha, beta, gamma and delta denotes the real and positive parameters related to the interaction between prey and predator species. Lockable-Terra predation equation is based on the assumptions. 1. Predator can eat limitless. 2. Supply of food resource i.e. prey depends on the prey population. 3. The rate of change of population directly depends on its size. 4. Environment is constant, inconsequential genetic adaptations for both species. 5. All-time unlimited food supply for prey. The equation is continuous and deterministic, indicated continuous overlapping of prey and predator population. The rate of growth for predator population is given by equation. dt equals delta xy gamma y. The change in predator population equals to food supply mediated growth minus death of predators. The growth of predator population is represented as delta xy. Delta is different from beta, as the rate of predator population growth is very much different from the rate of predation on prey. Gamma y is the decay rate of predator either due to emigration or mortality. In the prey absence, the predator follow an exponential decay. The solution to equation is periodic and yields a simple harmonic motion where prey population is traced by predation population by 90 in cycle Fig.3. The graphical presentation of Lockable-Terra model is given in figure below. 7. Figure 3. Graphical presentation of predator-prey interaction in model given by Lockable-Terra. A. Harmonic motion of prey-predator system and B. The combined zero isoclines of model by Lockable-Terra. Figure 3. Graphical presentation of predator-prey interaction in model given by Lockable-Terra. A. Harmonic motion of prey-predator system and B. The combined zero isoclines of model by Lockable-Terra. Source. %20 formatted.pdf. The prey isoclines are zero growth curves are horizontal and the predator isocline is vertical. Both isoclines intersect at right angle. Later, 1935, Nicholson and Bailey recognized the lacunas in the model of predator-prey interaction proposed by Lockable-Terra and developed a model for a host-parasitoid interaction. The Nicholson and Bailey's model involves paired equations. HT plus 1 equals lambda H1 express apt PT plus 1 equals HT 1 express apt, where H and P represent the host and parasitoid population respectively. Lambda is the finite rate in host population increase. A is the rate of parasitism per predator or parasitoid and T is the time. The early models given by Lockable-Terra provide insignificant biological realism and proved unrealistic and thus replaced by other models. Berryman 1992. In 1963, Rosenzweig and MacArthur developed graphic models which were regarded as best general 8 model. In contrast to the unrealistic horizontal prey isoclines of model given by Lockable-Terra, Rosenzweig and MacArthur provide more realistic hump-shaped prey isocline. Figure 4. Graphical representation of hypothetical prey isoclines given by Rosenzweig and MacArthur in Lockable-Terra model. Figure 4. Graphical representation of hypothetical prey isoclines given by Rosenzweig and MacArthur in Lockable-Terra model. The prey curve is convex in Rosenzweig and MacArthur model whereas linear per se Lockable-Terra model. Prey isocline is a fixed boundary at which the rate of increase in the population of prey is 0. It's a boundary between two phases, a zone of prey decrease and a zone of prey increase. The prey population size lies somewhere on this prey isoclines when established at equilibrium. Lockable-Terra assumed the prey isoclines as a horizontal line but Rosenzweig and MacArthur suggested more realistic hump-shaped prey isoclines Fig.4. The hump in the isoclines is caused by the limitation in the increase in prey population. The prey population is constrained by the ecological factors such as diseases, food shortage nor social interactions that limit their own rate of increase in population size. On the right side of the hump, the dominant limitation is the intraspecific competition between prey populations that causes the isoclines curve to fall off whereas on the left side of the hump, predators limits the increase in prey population. The upper limit of prey abundance and the exact shape of the isocline curve are determined by the carrying capacity of the environment and the demographic characteristic of prey. 9. Likewise predator numbers are also limited by certain ecological factors at high predator densities. When the prey density is low, the predator population is food limited as it depends on the population size of prey. But when the prey population is high, the predator population is limited by their own rate of increase such as territorial behavior etc. Resulting into predator isoclines Fig.5. Figure 5. A hypothetical vertical predator isoclines of Lockable-Terra model. Source. %20 formatted.pdf. 10. Figure 6. Rosenzweig and MacArthur model with predator isocline. Color line zone depicts the predator increase in abundance Fig.6. Vertical isoclines of predator assumed by Volterra suggested that predator population increases only at single prey density. A right minus bend predator isocline given by Rosenzweig and MacArthur. The isoclines is inclined towards right because as the population size of predator increase, intraspecific competition for breeding sites and other resources. The shape of predator zero isocline is not same for all predators. Moreover, in case of more efficient predator, the predator isocline shifts to left. The graphic model developed by Rosenzweig and MacArthur for the predator-prey interaction is given by superimposing the two isoclines, prey isoclines and predator isoclines. Thus, the effects of simple changes to the system of predator-prey interactions can be explored by using graphic model of Rosenzweig and MacArthur model. The model of Rosenzweig and MacArthur ignored the ratio of prey to predator numbers. To take into account this ratio of prey number to predator number, Arditi and Berryman hypothesized a ratio-dependent model Arditi, 1991. This model proposed that the rate of predation is dependent upon the ratio of predators to prey. As the prey abundance increase, 11. The predator productively increases and conversely as the prey abundance decreases the predator productivity decreases. Thus, predator and prey equilibrium both rises with increase in the prey abundance whereas fall with decrease in prey abundance. 4. Predation-prey system. The behavior predictions of predator-prey system given by Volterra model stimulated experimental investigations. Laboratory studies. In 1934, Gauss, the Russian biologist under constant environment conditions reared together a predator and prey ciliates, Didinium nosudum and Paramecium caudatum, respectively. He performed two different types of experiments in a simple environment. In one experiment, he introduced predator in a test tube containing clear nutritive medium supporting a population of Paramecium prey. Paramecium was unable to escape from the predatory ciliates Didinium, the prey succumbed to predation. On predation of complete prey population the predator, Didinium died due to starvation Fig. 7. Thus, from his experiment, he discovered that the predator always exterminated the prey irrespective of prey and predator population size. Gauss discovered that the predator population can be maintained only by periodically adding immigrants or prey to the medium. 12. Figure 7. Prey-predator system between Paramecium and Didinium in oat medium with sediments. Source: CS%20%20 formatted.pdf. In another experiment, Gauss introduced predator into the test tube containing sediment at the bottom acting as a site for prey to refuge from predator. Following consumption of all the available prey the predators died from starvation. Meanwhile, prey that refuge in the sediments increase in number and eventually spread over the medium. By periodically introducing predator to the medium, Gauss could be able to maintain both the population Fig. 8. 13. Figure 8. Prey-predator system between Paramecium and Didinium in oat medium without sediments with immigrants. Source: CS%20%20 formatted.pdf. Gauss's experiments did not support Volterra predation model and their predictions. Gauss concluded in his model that predator-prey interaction cause overexploitation of prey population followed by decline in predator population and the predator-prey system could be maintained only by periodically introducing the immigrants Fig. 9. Inspired from the work of Gauss, C. Huffaker 1958 established a complex laboratory environment to eliminate the possibility of self-extermination of predator-prey system. He introduced a mite, Typhlodromus occidentalis as predator and a six-spotted mite, Eotetranicus sexmaculatus as prey which feeds on oranges. Both predator and prey reproduces parthenogenetically. On trays, he established experimental population containing 4 rows with total 40 cells, 10 cells each row. He placed rubber balls in some cells whereas oranges of same size in remaining cells and thus regulate the pattern of food dispersion, total food availability to control the distribution and size of prey population. From his experiments he concluded that 14. A. For prolonged time, the low population size of prey leads to collapse in predator population. B. The immigration of prey is essential to eliminate the occurrence of self-extermination in the predator-prey system. C. The intensity of predation has relatively low influence on the period of oscillation in respect to predator searching for prey and prey dispersal complexity relationship along with the time required by prey to recover and repopulate followed by predation. Figure 9. Predator-prey system between Typhlodromus occidentalis predator and Eotetranicus sexmaculatus prey. Field studies. The field studies are supported by the laboratory experiments. Around 1839, an ornamental species of cactus called as prickly pear cactus was introduced into Australia from America. This species of Cactus opuntia, like other invasion species, escaped from cultivation and rapidly spread to nearly 60 million acres in New South Wales and Queensland. To control the prickly pear, a cactus-feeding moth was liberated from South America. The moth Cactoblastus cactorum multiplied, spread and removed the prickly pear plant until the cacti existed only in widely distributed small colonies Fig. 10. The decline in the plant also causes 15. Decline in moth. As the plant population increases, so did the moth until the plant colony declines again. However, few scattered seeds got established in new areas and formed new colonies to maintain the existence of species and predator-prey system. An unsteady equilibrium is maintained by the moth-cactus relationship wherein increase in the abundance and distribution of cactus plants subsequently trigger increase in number of moths and causes decline in cactus. These complex from laboratory experiment and field studies illustrate predator-prey system. Figure 10. Cactoblastus cactorum, moth on cactus and its larva feeding on cactus. 5. Functional and numerical response. In 1959, Holling suggested that as the prey density increases it leads to increase in predation rates due to 2 effects. A functional response where the consumption rate of predator increases in presence of high prey population density. B. Numerical response in which increase in prey density leads to increase in predator population density. 16. A functional response. There are 3 types of functional response curves which relate density of prey population to prey consumption by single predator per unit time. Type 1 response curve. It's a rare form of response in nature, for example in filter feeders. It's an initial exponential relationship between prey and predator, and this increase in prey consumption by predator continues till reaches saturation point where predator cannot eat maximum from that rate. Type 2 response curve. Type 3 response is also common in nature, depicting logistic increase in the rate of consumption on increase in prey density that level off to a plateau. This type of response destabilizes the prey population. Fig. 11. 3 response curve. It's a common type of response in nature, for example rodents and weasels. At low population density, the rate of consumption increases at decelerating rate, increases at slow rate, followed by sigmoid curve pattern of increase until it reaches asymptote satiation. The rate of consumption is dependent on two factors: a, the searching or locating a prey, and b, handling time of prey capture, kill and eat. At low population density, the searching for prey is important and predator kill prey at constant effort fashion, while at high density of prey, searching for prey becomes easy but handling time is limiting factor, and thus the rate of consumption increases slowly. Subsequently, searching is not required and rate of consumption levels off at maximum rate. It's a stabilized response attributing to the density-dependent amount of prey consumed. Fig. 11. 17. Figure 11. 3 types of functional response curves. Beat numerical response. Numerical response means increase in predator density on increase in prey population density due to direct responses, i.e., a, on increase in prey density the fecundity rate of predator increases, and b, aggregation response. Another type of numerical response may occur due to immigration of predator in response to the increase in prey density. A. As the prey population size increases, the consumption rate of predator increases. Leads to high fecundity rate and low mortality rate. B. Aggregation response. Predators' aggregation in prey hotspots is called aggregation response. This type of response increased prey-predator system stability. In 1966, Hassel proposed 3 basic types of numerical responses: a positive or direct response, in which predators' population increase in response to increase in prey density; b, negative or inverse response, in which the number of predators. This type of response increased prey-predator system stability. In 1966, Hassel proposed 3 basic types of numerical responses: a positive or direct response, in which predators' population increase in response to increase in prey density; b, negative or inverse response, in which the number of predators per unit area declines in response to increase in prey density; and c, no response, in which the predator population, unaffected by the increase in prey density, remains proportionately same. 6. Foraging theory. In nature, different biological factors such as intraspecific or interspecific interactions and physical factors such as landscape structure, etc., causes heterogeneous distribution of food. Therefore, in ecology the fundamental objectives persisted for long to study and understand 18. That how predators exploit food patches and effectively search for prey in the patches. Keeping in mind this objective, sequence of optimal foraging theories was developed by several ecologists. The optimal foraging theories provide basis for interpreting complex predator-prey interaction via testable hypotheses. The assumption made by the optimal foraging theories was that the predation activity is optimized to maximize the rate of net energy gain. Among various biological theories on animal behavior, the most extensively developed optimization theory is the optimal foraging theory, OFT. A patchy area is an area with high resources compared to average environment. Assume that the prey on which the predator feeds upon is distributed in the patchy area, then the predator spends its foraging time in searching and traveling between patches and rest in handling prey within these patches. Furthermore, while the predator searches for the prey, predator depletes the number of prey within the patches, and gradually with time the benefit of staying in that patch declines. According to optimal foraging theory, the predator may face 4 types of difficulties: a, the choice for optimal diet; b, the choice for optimal patch, what type of patch should be searched; c, optimal departure time from the patch; and d, choice of optimal movement between patches. In order to maximize the foraging efficiency of the predator should make decisions assumed by the optimality approach as per the optimal decision rules. 7. Summary. When one organism consumes another living organism, the interaction is called as predation. It's a type of plus or minus relationship where one individual is benefited at the expense of other. In the broadest manner, the predation includes parasitism and herbivora. The predator-prey interactions of organism with discrete and continuous generation systems have been described by the mathematical models. The continuous generation model for predator-prey interactions is given by many ecologists. The very first but unrealistic model is given by Lotka-Volterra, followed by more realistic models given by Nicholson-Bailey, Rosenzweig-MacArthur, and Arditi-Berryman model. All these mathematical models in ecology predicted 19 and concluded the oscillations of prey and predator population. The resulting oscillations from the interaction may be unstable, damped, or stable. The predator-prey relationship causes 2 types of responses, namely functional response and numerical response. Predator may consume more number of preys on increase in density of prey population, called as functional response. Predator may increase in number in response to increase in prey population, called as numerical response. The functional response can be of 3 types: type 1, type 2, and type 3 response. In type 1 functional response, the number of prey consumed per predator is directly proportional to the increase in prey density until it reaches maximum. In type 2 functional responses, as the prey density increases, the number of prey consumed by the predator increases at a deaccelerating rate until reaches maximum. In type 3 functional response, the predation rate increases slowly as the prey density increases, later following a sigmoid curve until reaches asymptote. Numerical response includes aggregative response, in which as the prey density increases it attracts the influx of more number of predators to that area. Furthermore, numerical response also refers to increase in the rate of growth and development, natality and mortality rate alters of predator population. The food is distributed non-uniformly in the habitat resulting into the patches where prey occurs. Thus, in terms of profit relative to net energy gain, the predator spent time in hunting prey occurring in patches with more profit rather than patches with abundance of prey. This behavioral study of predators leads to development of concept of foraging theory. The optimal foraging theory (OFT) is a strategy adopted by the predator to consume its prey in such a manner that it should gain a maximum rate of energy. The optimal foraging theory includes the choice for optimal diet by the predator, the choice for optimal patch, what type of patch should be searched, C. Optimal departure time from the patch, and D. Choice of optimal movement between patches. 20.